Greetings, everyone. In this video, we're going to be continuing with the PCR sequence analysis. In the previous video, what we have managed to achieve was to go from our AB files that had our chromatograms to a consensus gene sequence file in FASTA format that had the resultant of both the forward and reverse sequence reads. Now, in this video, what we aim to do is take that resultant sequence read and query that sequence with sequences that have been deposited on a free and open gene bank, such as the NCBI GenBank, and analyze our gene through methods that involve that. So this is our concatenated consensus fly cytochrome oxidase gene sequence. We know what gene it is because of the primers we use during the PCR stage. So we know, since we know the gene identity, all we are doing is basically try and figure out how can we identify the fly that we have based on the sequence. So obviously now we are taking a scientific approach. So the presumptive ID is a green bottle fly species, but there's many different types of species that this could be associated to. Most green bottle flies belong to the genus Lucilia. However, species ID is required. So basically, by analyzing this gene, we'll be able to get concrete ID on what this fly species is. So that is kind of the question we are going to address in learning how to further analyze this consensus gene sequence. So once you have your consensus gene sequence, what we're going to do is we're going to copy the sequence and we are going to query it on the National Gene Bank. And the site we're going to use is BLASTN. We can find the site by just typing BLASTN on Google. And it should be the top option, the first selection that you get. So the N in BLASTN stands for nucleotide. There's various different types of BLAST. BLAST is basically one of the terminology used by the NCBI GenBank to say give me your sequence and I'm going to search it. So we have BLAST for different types. We have BLAST for proteins as well. But since we're working with a DNA sequence, we're going to be using BLASTN. And all we're going to do is we're going to paste our FASTA sequence of the consensus green bottle fly cytochrome oxidase gene into the search box. And we are going to query this sequence with sequences that have been deposited on the GenBank. So now what we're going to do is we're going to look through the BLAST parameters. So what we need to make sure is under choose set search, it is set to standard database. Remember, we're working with DNA of a organism. So obviously now we are not working with RNA. We are not working with genomic DNA. We're also not working with any viral components. Let's also make sure that the organism block is empty. These models should be left empty. And under program selection optimize for, we always select highly similar sequence. There may be some rare instances where you would want a less stringent search query, but those situations are very rare. So once you have inputted your parameters, you would click BLAST and your sequence would be queried. And this could take some time. So I've done this in hindsight. So I've done this beforehand. So when your sequence is finished being processed via the GenBank algorithms, a window like this would appear. So it would have the name that your consensus sequence was labeled. In this case, we labeled ours as a green bottle fly. It'll tell us our query ID, it'll tell us the description, it'll tell us what molecule type we're working with, it'll tell us the length, and it'll also give us the description of the organisms. that the sequence has showed the highest degree of similarity to. So these are all the descriptions, so basically the names of the sequence files that our queried sequence has the highest degree of similarity to. And adjacent to that on the right, we have the name, the common name, the tax ID, the maximum score, queried score, e-values, percentage identity, and excision numbers. So of these, we generally focus on the e-value, e-values of 0 or pretty close to 0, like we're looking at any number times 10 to the negative whatever. Very close to 0 or 0 is good because it tells us there's a very low chance of error that we got a false assignment. Then we look for stuff like percentage identity. So how much does our sequence match the hits that we got acquired? So basically, these scores, you can look it up on Google for better understanding of what they represent. In this case, we are going to focus on the percentage identity, we're going to focus on the e-value, and we're going to focus on the accession. So the reason why the accession is so important is if we are going to say that our sequence, our cytochrome oxidase sequence from our fly matches these the corresponding these corresponding species, we need to show for reproducibility the excision numbers of these hits so they can be tracked back in case someone needs to query them. Now, we cannot work with all the gene sequences all the sequence hits that the BLAST search has given us, because then we'll have too much of data. So in this example, we're going to cut out a huge chunk of this, and we're going to just limit our analysis to basically 10 sequences. So I'm going to take the first 10 hits, and the criteria I'm using is I want a query coverage of anywhere greater than 98%. Normally higher is better, but we do get some instances where ID is a bit lower. I want a percentage identity of greater than 99%. I want an e-value of 0. So as I said, the more stringent you set your assignments, the better. So query coverage of greater than 99% would be the best. So that's in the name of good practice, let's stick with that. So we're going to select the first 10 or the top 10 hits that have a query coverage of greater than 99%, an e-value of 0, a percentage ID of greater than 99%. And luckily for us, those parameters are satisfied within the first 10 sequences. 10. Okay. So once we have selected the sequences that we want, we now need to download these sequences. And we do that by hovering over the downloads pane. So we have selected the first 10 gene sequence hits that have the criteria we want, and now we're going to download them. We're going to click download. We're going to download the FASTA aligned sequences. And what we're going to save these files in the same folder. Everything has been saved as previous. And within this folder, we're going to make a new folder called sequence hits from GenBank. Now, we all can see that GenBank is saving this under the file name sequence dump and saving it as a text file. Now, since majority of our software that we use is going to analyze gene sequences as either a FASTA file going forward or as a .meg file. So we are going to rename this file so it is more intuitive to us what it might be, and we're going to save it as a FASTA file. So since this is the sequence hits from GenBank, we are going to label it as green bottle fly sequence hits from GenBank. And to make this a faster file, we're just going to end this off by adding a dot and typing FAS. So when this is saved, it will be saved as a faster file. We also need to save the description of the top 10 hits. And we're going to do that by going to downloads and saving them as a descriptive table.csv. And again, GenBank is going to give it its own assignments, name assignments. And what we are going to do is we are going to relabel it so it is more intuitive to us what it might be. So this is going to be the description of hits the description of the hits from the green bottle fly query. And we can see it saved and saved in a format that Excel can recognize. So once we have downloaded the sequences that our sequence corresponds to and downloaded the description of what the hits are, we do not need to work with GenBank any longer. And what we're going to do is we can close our web browser, we can close BioEdit where our file was, where our consensus sequence was. And now we can go into the sequence hits file and look at the hits that we downloaded. So we're going to open this file with BioEdit, and we can see, since we have downloaded the FASTA file containing the aligned sequences, the sequences that have been downloaded are already aligned. And if we expand this further, we can see in this column where we generally have the name, we have the description, the full description of the sequences that appeared on GenBank. Now, this is quite long and it has a lot of unnecessary information for us. So what we're going to do is we're going to trim the names, and the bits of information we are going to keep would basically be the genus, species, and strain. We don't need to know that it is a cytochrome oxidase gene because that is the gene sequence that we have targeted. So all we're going to do is we're going to leave the strain designation. If it says voucher, that is not important. So we have left the genus, which is Lucilia, the species, which is Cuprina, and the strain, which is LC MUAR-2. And we are going to leave the excision number. We do not need the lengths starting and beginning. All we need is the excision number, because when we do our phylogenetic analysis, or when we do our phylogenetic analysis, and when we are displaying or presenting our phylogenetic tree, what's important to know is what genus and species were our hits. What strain is not as important, but it gives a good idea that this was a different strain, or a different strain of the same species or different species. And the excision number tells us if we needed to go back and double check, this is the excision number we need to look for when we are double checking. Now we are going to go through each of these files and we are going to edit them in the same way. So once you have finished editing and polishing up your sequence hit names, just do a final check. All the excision numbers, you have the genus, you have the species, you have the strain. I have included whether they say isolate this or isolate that, just so, because sometimes the isolate names can have some confusion associated with it, especially with higher organisms. With microbes, it's much more simpler. Anything after the species, unless it says subspecies, It is generally the strain. With higher level organisms like flies, insects, and stuff, it can get a bit confusing. So I'm going to leave these as is. So this is our processed bioedit file with our hits. Now what we want to do is we need to include our sequence that we queried for comparison. So to do that, we're going to open the consensus sequence of our fly. We're going to copy this and paste this to our blast save, our bioedit table that has our sequences, and we are almost good to go for further phylogenetic processing. Now, in many cases, in phylogeny, what we need to have, the three main criteria that we need to have for proper taxonomic study via phylogenetic analysis is that we need to have our queried sequence. Our sequence. We need to have the sequences that it's most likely related to, and we need to have a sequence that the software can use to root or set a baseline for comparison from. But since we are working with insects, so once we have polished up the BLAST hits and copied our consensus sequence into these, and even and sometimes you can even get a bit more if you know a reference cytochrome oxidase sequence of a bee, and you can very easily just search examples of just basically the name genus of a candidate bee, and you'll be able to find. And if you search that on GenBank, just input the name, and you can put cytochrome oxidase, type nucleotide, select the nucleotide option and search. You will be able to find the cytochrome oxidase sequence for many species, for many of the species you queried, and some may be a bit different. But you will get a large majority of cases what you are looking for. Now, many organisms have been identified via the cytochrome oxidase gene. It's a very good gene target for identifying eukaryotic organisms. So in our case, we have got our reference root, and just be careful. There are some that are targeting, some may target different cytochrome oxidase regions. In our case, our query seems to be fine. So I'm going to download the sequence that has the highest linear DNA sequence. In this case, I'm going to choose the second option. I'm going to select it. And the whole purpose of doing this is so we can download the FASTA file of its nucleotide cytochrome oxidase subunit 1 gene sequence. So if you're working with other genes, make sure, okay, while it's loading, I'm just going to click FASTA to help direct it. So when you're working with other genes, if you're working with, in this case, you're working with the cytochrome oxidase subunit 1 gene sequence. If you are downloading the FASTA and you are downloading a different subunit, you are not comparing like things together. So make sure your gene is the same and correct, and make sure your the subunits, if you are targeting a certain subunit, is also the same. All right. So once we got the gene sequence of our root, we are going to download this file. So we're going to click send to. We're going to download as a file, as a FASTA file, and we are going to rename this as. We are going to rename this as the name of our root. Singular. And in this instance, GenBank has saved this as a FASTA file. So that works out fine for us. We can, once the file is downloaded, we can open it, copy this sequence into the same document. We can copy the sequence into the same document as our last hits and our code sequence. And we are also going to trim this as how we have done previously. Once we are done, we're going to save this. And we are going to make an extra copy of this. And we are going to save it in the same folder in our desktop. But now we're going to make a new folder called Mega Phylogenetic Analysis. And we're going to save this as a FASTA file in this folder. So now when we open this, we should have our sequence file here. Now that we have a single file that has our consensus sequence, it has our queried hits, and it has our root, we are going to open this in Mega. So I'm going to start Mega. I'm going to direct Mega to the location of the file. And if we have everything in one file, it's very easy to navigate and direct to. Mega is going to ask me, do I want to analyze or align? I'm going to click align. And as we've done previously, I'm going to select everything. Control A is quick to select everything. I'm going to align them by muscle. And very quickly, Mega has aligned our sequence. So our root, so as you can see, our root does not match up identically with many regions of our queried, of our GenBank hits and our sequence, but our sequence that we have queried matches up lovely with the hits that we got. So if you see something like this where your root breaks up, it is normal. However, if you think that this sequence could have been submitted in the reverse direction, what we can do is, as a check, we could reverse complement the root. Align it again. And if nothing really much has changed, you could then assume that it's just due to the variability in the gene. So I wouldn't say in all cases, but depending on the genes you target, so I've just basically undone the reverse complement that I've done. So this is in its native form that we have downloaded from GenBank. So in many cases, your root will not have such a high degree of variability like this between your queried hits, but it is not uncommon for certain genes and certain roots to have such variability. And if you want something that's more conserved, you could look for a sequence or for an organism that's a bit closely related, but has some noticeable differences. So what we're going to do now is, since we're going to do a phylogenetic analysis, what we want is all of them to have the same starting point, and all sequences that we have yet have the same starting and ending point. We do this by trimming the overhangs. They're at the beginning and at the end. So here we see from this point, if I hold Shift and click, the region highlighted in yellow has regions where there are gaps. And we want to delete these regions so all of them start at a point where they have the same, where a nucleotide is basically occupied. So I have selected the region of overhang and I'm going to delete it by just clicking delete. I'm going to go to the end of the sequence. I can click and then hold shift and click to designate only this region where the overhangs are located. And I can delete that. So basically all of them have the same starting point that is occupied by a gene, by a nucleotide. I'm going to export this file as an alignment, and I'm going to export it in a mega format. I'm going to save this in my mega phylogenetic analysis file, double check that it is a mega file or a .meg file. And you could give this data a title. I'm going to call it this phylo analysis of insect cytochrome oxidase 1. And once that is done, we can close mega and we can open the file we have just stored, just saved. Remember, it's in a mega format, so we can just double click that and mega will open. This TA part has our data. And what we want to do is click the phylogenetics option and begin our phylogenetic analysis by constructing a maximum likelihood tree. Mega is going to ask you, would you want to use the active data set that is open? You're going to click, well, for me, this will always be, I'll always use the active data set, and I'll click yes. The statistical method for analysis is the maximum likelihood. The phylogenetic test will be done via the bootstrap method. If this was not selected, we would select bootstrap method, and the number of replicates in our lab, the standard is 1000. And then we're going to leave everything else as is or as displayed over here. We're going to click OK, and Mega will run the phylogenetic analysis. It will repeat this analysis 1,000 times because we have designated that the bootstrap should be done for 1,000 times. And when this is done, we will continue the video. Okay, so Mega has finished analyzing the various hits and the consensus and our root, and has assigned phylogenetically the relationship between our sample and the closest and the GenBank hits of it. So the phylogenetic tree would look something like this. And what we can see is our root is clearly the outlier, which is what it's supposed to be. Here's our sample, here's the hits. And what we need to do now is tell Mega which is the root. We do that by selecting the subtree and clicking root, and then clicking the sequence, which is our root. Now, once that is done, we can select two. So once that is done, we can then infer phylogenetic relationships or taxonomic identification of our species versus our sample versus its hits. And from this, what we can see is that the GenBank hits seem to be related to, but phylogenetically distinct from our green bottlefly species. They are, which tells us, so when you see something like, download this or export this as a file that we can use. I prefer saving mine as TIFF files, but some computers, I'm not too sure why, but some computers not like to files as much. So in this example, we're going to be saving it as a PNG, and we can give us a file name. So I'm going to call it phylogenetic tree of green bottlefly. I'm going to save this in the same folder where all our molecular phylogenetic stuff is stored. And I'm going to call it again phylogenetic tree of green bottle fly, and I say it as a PNG. And this can be copied into someone's thesis or paper and be used. So this is a quick video on how to do the phylogenetic analysis. And another point to remember is these sequences came from GenBank. So it's good practice to include, it is good practice to include in your write-ups, in your thesis, a summarized version of the descriptive table where you got the hits from your query. Bye.